I'm going to give you, it's going to be a heroic effort to summarize give you a high-level talk on low-level analysis. So try to explain to you what are the hurdles that we have when we think about how to normalize and how to work with technical variability in single-cell RNA-seq data, what are the current efforts, and where are our challenges. So, all right, I guess I can just use this. So when we look at single-cell RNA-seq data, as some of the speakers before me talked about, we have all of this amazing variability that we can see, and we can learn new things about biology from it. But we need to be very careful when we look at this data as it comes out of the sequencer. And the reason is that in addition to the biological variability that we have between single cells, an additional component that really dominates the variability is the technical components. And technical variation. When we talk about technical variation, I kind of laid out here a list of a few of the factors that are what we call a technical variation. So the first thing that we have there that we need to think about is that we have many RNA-seq libraries, and each one of them can have different quality and different depth to it. Another component that is very strong, confound single-cell data quite a lot, is the fact that we have limited sensitivity. And that means that we can miss a lot of genes, and in some ways we can also meet a lot of interesting cells. Some more idiosyncrasies for single-cell data is the presence of doublets and dead cells in our wells, and some enhanced effects of gene-dependent biases, and of course, the everlasting batch effect. So where is it all coming from? So I'm kind of trying to think about all of these factors and which parts of the technologies give rise to them, right? So basically, we're trying to think about what in those technologies distort the picture from the biological data to the files that we end up seeing. Now, it's kind of hard to make generalizations when you look across different technologies. There are some parts of them that are more important, some parts that are less important for this issue, and they are different in many ways, so it is hard to make generalizations, but I just summed up with the three main contributors to these technical variations. So the first one has to do with basically taking the cells from the biopsy to capture and to lysis, and when we do that, it can create things like biases that are related to cell properties, like the size of the cell, the position of the cell, or the timing that it took basically from getting the cells until lysis. It can also introduce some variation in the amount of liberated RNA, which already introduces some noise. The second part has to do with mRNA capture and RT. So again, in this part, we can have quite a lot of variable efficiencies, for example, because of bead to bead variation and limited sensitivity that gives rise again to the limited coverage that we get in single cell data. And finally, everything that has to do with amplification bias, things in PCR amplification that create bias in things that are gene-specific properties like GC content and length. Of course, this is addressed to some extent when we're working with UMIs. Okay, so this is the problem that we have. We have all of these confounding factors, and this is just to convince you that these confounding factors are indeed important. So when we have single-cell RNA-seq data, FASTQ files, we can take them and then we can extract from these files a very long list of parameters or features that can give us some indication, some proxy for these different quality issues that I talked about before. So here I laid out some of them, and some of them have to do with the depth of the library. Some of them have to do with alignment statistics. Some of them have to do with possible gene level biases that we have in the data. And the bottom line, what I want to show you here, so we have two sets of parameters here. One is for 10x drop-based data with UMIs. The other one is for the SMART-seq2 protocol. The bottom line is that whichever data you're looking at, if you take the data as it is unnormalized, these factors will confound the data very much. So what we see here on the left, this is 10x data on T cells from humans, about 4,000 T cells. And if we take these quality metrics and we correlate them with the first principal components, we see very high correlation, which means, for example, that the first principal component can be almost entirely explained by depth. And this below here is a TSNE plot of the same data. Similar thing we see in SmartSeq2. So in this picture, we see some data from mouse T cells of about 300 cells, much better coverage, but still, if the data is unnormalized, it is very strongly confounded by these technical factors. All right, so this is the problem. Now, to make this problem even harder, so here's the problem. We cannot simply remove it. And the reason we cannot simply remove it is that a lot of these confounding factors are confounded with biology. So this looks a bit like an ice cream, but it's supposed to show you that we can have like different cell types that go together into one analysis, right? So the first thing that kind of a cell intrinsic property that is important but can give rise to libraries of different quality that we don't really want to normalize is the size of the cell that correlates with the RNA content of the cell, that we correlate with how many genes we can detect and how good the library is going to look like. So here we need to be very careful. These are some results from a recent review that John wrote, where we can see that as you go in cell cycle, the RNA content of the cell increases or decreases as you follow development. Some more speculations about things that can contribute, so biological properties that can contribute to something that will look like technical variability has to do with the milieu of genes that are expressed by the cells. So for example, in some cases, the cells can have tendency to express more transcripts that are short. And this is one example. If we, for example, look at transition, the T cells go from oxidative phosphorylation to glycolysis, which is very important. It also comes together with a reduction in the average transcript length, which can give rise again to libraries that look like they are not as well and not as good. And this is something that we don't really want to correct when we do normalization. Okay, so we have this problem now, right? So we have all these confounding factors that are confounded with biology, but we want to normalize for it. We want to eliminate these technical factors. So ideally what we want to do is we want to have some technical standards. We want to have some things that are invariant. We can put them in our libraries and use them for control. Of course, the problem with single-cell RNA-seq data is that there is no really a notion of a true technical replicate. We cannot profile the same cell twice. But instead, the approach here is to use these kind of invariant standards that are added to the cells. And one type of these are, as many of you probably know, are the spike-ins, which are basically exogenous RNA molecules that come in predefined concentrations that we introduce to the cells during lysis. So basically, every cell has every cell has these RNA molecules that we know how many of them should be inside the cell, and then we can use this information to basically estimate the total RNA quantity in the cell and also things that have to do with technical variation, at least post-lysis. Another cool technology that was introduced quite a while ago, but not really, it didn't really catch up too far as the pull and split. But basically what we do, we take, in this case, we take, let's say, 10 cells, lyse them together, and then separate it in the mix into 10 wells. So, and then the differences between these wells should reflect some sort of how does technical variability looks like, and this can be taken into account in an analysis. Now, the problem with these, so these two technologies are helpful, especially the spike-ins has been used quite a lot. But the caveats here is, in the spike-ins, the most popular one is the ERCC collection. The problem with this one is it's been shown to be maybe not too reliable to vary between technical replicates, and also the properties of these RNAs are intrinsically different from the properties of RNAs that are endogenous in terms of their GC content and in terms of their length. But maybe more importantly, both of these technologies, especially, I guess both of them, it is still debatable to what extent they will be useful or they can be convincingly embedded into the key technologies, especially the drop-based technologies. Right? Okay. So this is our problem so far. So we have all of these confounding factors to the data that are confounded with biology, and when we go to technical standards, there are some ifs and buts there. Right, so we do need to do a normalization here, and this is how a general normalization pipeline is going to look like. There are many steps here that have to do with scaling, with handling with dropouts, and mitigating some additional confounding factors. I'm going to talk today only about two of those, about global scaling and about gene-specific corrections. what is global scaling? So here the idea is very simple. Basically, what we want to do is we want to take the expression of a gene and decouple it to the observed expression of a gene and decouple it to the size of the library, some contribution from the cell, and the real expression of that gene, right? So from the bulk era type of methods, of course, the simplest method would be to take every cell and just divide the reads by the number of reads that we have totally in that cell. This is, of course, can be a bit sensitive to transcripts that have very high counts in them, and there are more robust methods like DSeq and other methods that go kind of for the median and give us nice and robust scaling factors. The problem with these scaling factors, the first one, the problem with applying them to single-cell data is the first thing is we have zero inflation. We have a lot of zeros in single-cell data that can make some of these methods not so useful. The second problem is that these methods make assumptions, especially about the fact that our samples, the number of differentially expressed genes is not very high, is less than a half, which is not necessarily the case in single-cell data. And this is, again, a result from the same review from John and colleague, John Marioni and colleagues, that basically what they've shown is that if you use these methods, they can give rise to estimations of scaling factors that are either overestimation or underestimations of these factors. So going to the single-cell normalization factors, so we have kind of two families of these algorithms. The first one is based on spike-ins, kind of taking the information from them, the variability between different libraries, in order to derive scaling factors. The second one is kind of a neat approach, which is basically pool cells together. And this basically gives you a way to do two things. The first thing it gives you is a way to account for the dropouts, because if I have a zero in one cell, but in the neighbors it's not a zero, so I can still use that to do scaling properly. The second thing is that it can be aware for the presence of subpopulations, so I'm not basically having like one big scaling factor, but I am sensitive for maybe the presence of cells with larger RNA quantity versus cells with lower RNA quantity. And if you look at the same simulation data, this is this method on the right, it's called SCRAN, and you can see that the scaling factor that it infers are actually more correct. But the thing is global scaling is enough, basically taking the RNA-seq, the count result or the UMI result and just dividing it by a single number. This is nice, but in some cases it is not enough. After we divide by this one number, we can still have a lot of confounding factors in the data. And this is one result from a recent paper that came up that shows that even after scaling, in some genes, in many genes, we still see a correlation between the scaled value of the gene and the depth of the library. And we can see the same thing. This is the test we've done in my lab. We can see also the same thing in 10x data. To make things even more complicated, there are other confounding factors that go into the data that are very, very hard to eliminate only with scaling. And this has already been shown in the bulk level data. One of them is batch effects. And we can see here, again, the T-cell data. If we scale it, we still see a very, very strong batch effects. So how do we address this? Oops, I'm already running out of time. So yes, so how do we address this? So here is basically how a model would look like. So the idea here is basically to take every gene and model it, decompose the expression that we see by contribution of different effects to the gene. And these are the different contributions that you can see here, all the different combinations between known effect and desired effect, unknown, undesired, undesired, known, and so on. So basically, we take every gene and we want to take the contribution from these different aspects to that gene, right? So known wanted, for example, would be what is the biological condition. Known unwanted would be batch effect or quality factors. Unknown and unwanted are things that we can derive from the data using, for example, negative control genes, housekeeping genes. And unknown wanted, again, are things that we can derive from genes that we think that there is interesting variation going on there. And all of these things can be kind of implemented into a model. These are two examples for such models. We have Scatter, which uses the composition of the mean, and SCLVM, that uses the composition of the variance. But the idea is they say, basically, take the data and decompose it to these components, and then only leave the ones that we care about, the ones that we want to work with. Now, in the interest of time, I'm going to skip this. And just end up with saying that there are many, many ways that we can normalize data. And there are many different considerations of how we want to build these kind of models. We can scale or we cannot scale. We can take a given scaling method or another scaling method. When we build a model, we can take any different subsets of these different covariates, known and wanted, unknown and unwanted, and so on. So the question is, which one should we take? So the reality of data normalization is that there is no one method fits all. This is something that we've been working on, trying to think about how to do that. And this is a method called SCORN that we've developed. And basically, the idea is to flip the problem on its face. And instead of trying to develop a normalization method, what we've done is to develop a way to evaluate how good a normalization method is. So the idea in SCORN is to consider many different normalization methods and come up with ways to evaluate them, which gives us at the end something that can be a good and robust normalization. I'm just going to skip to the end here and say that in summary, I showed you what are the challenges that we have. Basically, the big challenge to separate technical from biological variation. As we go ahead, what are the main challenges that we need to address here? So I think that the most important questions are how to address the unknown, the dropouts and the cells that we not observe in our data, how to identify doublets, and maybe the most important thing is how to think about best practices for normalization. And with that, I will conclude. Thank you very much.